In Section 5 and Section 6, we're going to be looking at the genome projects, and we're going to be talking about some of the sort of ideas on using genes to fix individuals. So the genomics involves the study of genes, the gene functions, the entire genome. So genomics is the study of the genome. And it can include sequencing the genome, comparing genomes within and across species. So gene sequencing is determining the order of DNA nucleotides in genes or in the genome. The genome of several different organisms have been sequenced, as you can see right there. Even humans are in that category. The Human Genome Project was a project that its job was to sequence all of the DNA base pairs of human chromosomes. And we ended up having that done several years back, and we're able to analyze DNA from a few people. There's a couple of projects out there where it's like the 1,000 Genome Project, where they're taking it from thousands of different people and processing their genomes, being able to use it as a database for that. So we're still working to identify and map the different human genes. So technology has allowed the study and comparison of both genes and proteins. Bioinformatics is the use of computer databases to organize and analyze biological data. DNA microarrays are used to study the expression of many genes at once. And that's what you see on the screen right there is the microarray. So what you would end up doing is you'd have these little glass or silicon discs that would have gene sequences on it, and then you could take genes of an individual that you're wanting to see. Do they have this gene on in them? And on those discs, they're single-stranded, and you'd end up making those genes single-stranded, and you add them to it, and you'd be able to see whether or not like it turns green or red. Green means that the gene is active, red means that it's not active, and yellow means that in some areas it's on, some areas it's off. Another thing is proteomics, and that is the study and comparison of proteins. So basically figuring out how the proteins fold and bend and how they're put together is the next sort of big step, is figuring out how that process works. So genetic screenings can detect different genetic disorders. So genetic screening involves the testing of DNA. It's going to determine the risks of having or passing on genetic disorders, and they're used to detect specific genes or proteins, and it can detect some genes related to increased risks in cancer, and can detect some genes known to cause genetic disorders. So you could do it pretty much like that microarray thing for some of it, or you could end up doing a DNA fingerprint type of a thing and seeing, do you match with these areas that of individuals that have this genetic disorder. Gene therapy is something else that can be done. Gene therapy is the replacement of faulty genes. So gene therapy replaces defective or missing genes or adds new genes to treat a disease. Several experimental techniques are used for gene therapy. Genetically engineered viruses are used to infect patient cells, and that virus will insert the gene to stimulate the immune system to attack cancer cells. You can do that. you can insert suicide genes into cancer cells that activate a drug. So it basically will say you need to turn this on, and it ends up doing that, and it kills the cell. Gene therapy has many technical challenges to it. Inserting gene into the correct cells is a big thing. Controlling gene expression is another. And determining the effect on other genes. So I will talk about this a little bit in class on the gene therapy and some stuff that may be sort of questionable. So that is the end of the notes for section 5 and 6, and end of the notes for chapter 9.